Without any further ado, I'd like to introduce our speaker for today, Dr. Martí Català Sabaté, who is a senior data scientist who uses routinely collected healthcare data to generate reliable evidence for improved patient care. So he co-leads the Oxinfer group in the Health Data Sciences section of the University of Oxford. His research focuses on generating evidence on safety, effectiveness, and cost-effectiveness of medicines and procedures, where he develops R packages and code to curate and analyze data from millions of routinely recorded healthcare interactions. So Dr. Sabaté also contributes to the Darwin EU project as a programmer and study lead. So this project is from the European Medicines Agency and seeks to deliver real-world evidence from across Europe on diseases, populations, and the use and performance of medicines. Before joining Oxford, he completed his PhD thesis in computational and applied physics at the Universitat Valenciana de Catalunya in Barcelona, where he built computational models to understand the natural history of tuberculosis and also to predict the evolution of the COVID-19 pandemic, which were used by the Catalan authorities and the European Commission DJ Connect. So Dr. Sabaté is going to be going over his work on observational methods for COVID-19 vaccine effectiveness research and empirical evaluation and target trial emulation. So I guess over to you, Dr. Sabaté.
Thank you very much for the opportunity and for the introduction. As mentioned today, I'm going to present this methodological research that we did 2 years ago about observational methods for COVID-19 vaccine effectiveness research. Basically, it's an empirical evaluation and target trial emulation. So first of all, disclosure, like the study that I'm going to present was funded by a grant from the an NIHR grant. So here the reference. And yeah, thank you very much, Mauro, for introducing me. So just very quickly, like my background is on physics, so I didn't start with epidemiology, moved there with my PhD on tuberculosis and COVID, and now I'm working on the University of Oxford. Here you have the link to the group where you can see the website and what we do. Basically, we do epidemiological studies, some method research, and development of standardized tools for the Darwin EU and HDR UK project that you may have heard about. All the research that we do and the same that I'm going to present today. So it's done in the common data model and so. So yeah, what I'm going to present today a bit. I'm going to start with a bit of a background, why we did what we did, and which were the objective of the study. Then most of the time I will be speaking about the method research, this paper that I'm going to give an overview and like show all what we did. And at the end, in principle, we also have time for to explain about learnings, application, and reproducibility of this study. This study led to 3 more studies that were done like using the methods evaluated in this paper. So yeah, to start with a bit of the background, like well, explain about COVID at 2024. I don't think that it's needed, but yeah. So COVID-19 population kick in end of 2019. Then there was like a fast a big effort to develop like the vaccine, and there was like a massive vaccination campaign that showed like very good trials with different brands that were approved in different places of the world. And then it was, okay, we've seen in the trials very good results, 80 to 95% productive effect, even bigger in some cases, but which is its effectiveness in the real world data. And that's how this project started. We wanted to evaluate the effectiveness of the COVID-19 vaccinations in real world data. So then these are areas like how we can do it, and doing some research, we've seen, well, like doing vaccinated versus unvaccinated research is it leads like to many difficulties. One of them is which is the index date of the unvaccinated, and that's like we are going to try to see two different approaches to deal with this situation because we don't have an unvaccinated date. And also there are like many complications when we deal with this research, for example, like deal related to confounding different age comorbidities associated with like the COVID increased risk. So we have many different factors between vaccinated and the unvaccinated population that we want to minimize these differences. And usually like we do this with matching weighting, but there are like also many different ways to do it. So our main idea was, okay, we want to answer this question, which is the effectiveness of of COVID-19 vaccines in real world data. And we want to do some research on which is the best method to answer this question. So as I said, the goal, evaluate the effectiveness of the first dose versus the unvaccinated population. And to do that, we did first an empirical evaluation of 8 different methods based on overlap weighting, inverse probability, treatment weights, and matching. And then we conducted a target trial emulation, trying to emulate the results that we already seen in some of the phase 3 trials. So yeah, I'm going to start with the first part. We want to do this comparison, first dose versus unvaccinated. So we did that using the CPRD data base. CPRD ORUM database, it's a primary care data in the UK with around 40 million individuals. Around like there is a part which is like historical data, but like around 8 million individuals are still active right now. So this is going to be the population that we are going to work with. It's important to note that this database has complete linkage of COVID-19 testing and vaccinated status, and that's why it's good for the research and the purpose that we wanted for our question. So which is going to be our population of interest? We are going to pick individuals that were on observation 4th of January 2021, older than 75-year-old, and at least they were visible in the database for at least the 6 months prior the start of the study, this 4th of January. We needed also, we requested location to be present so we can explore some sensitivity analysis related to the location. And then we created 2 cohorts related if they were vaccinated or unvaccinated during these 4 first weeks of January. So we separated all the people who fulfilled the inclusion criteria. We separated them in 2 cohorts, the ones that were vaccinated between 4th of January and 28th of January, and the ones who were not vaccinated. Yeah. So then, so we've seen that was something expected. There is confounded, like the characteristics of the individuals that were on the vaccinated cohort were different to the ones that were on the unvaccinated cohort. The first more clear difference was related to age. So vaccinated individuals were older. That was an expected result. And also we've seen here, I just put like the first 3 conditions that we use to assess, but we have like in the paper, like a long table 1 of characterization, but we've seen that in the vaccinated individuals present more comorbidities than the unvaccinated ones. So that was an unexpected result. We have confounding in our study, so we have to deal with confounding. How we deal with confounding? Using like a common approach. There's like propensity scores that it's like the probability to receive a vaccine. Hopefully everyone is familiarized with propensity scores. And then propensity scores is what we use it in our study. So first of all, how we calculated our propensity scores. So we did a mix between key confounders and a data-driven approach. So for the data-driven approach, basically what we did is do like a large scale characterization of different conditions on drugs. For conditions, we use it like 3 different windows, non-overlapping, but the month before, the 6 months before, and any time prior. And for drugs, we use it only 2 windows, the month before and the 6 months before. From all these, we selected any condition or drug that any individual had, and to select them, we did like a 3-step inclusion criteria. First, we only selected the condition, the covariates that had presented more than 0.5% of in the study population. So at least they were quite frequent or a bit frequent. All these covariates were fed to were used in a lasso regression for variable selection, and we used lasso as a selector of covariates. And finally, with the resultant covariates, we did a clinical review to exclude like instrumental variables. Apart from the data-driven part, we used we forced some covariates there that we considered key confounders that were location, age, prior observation, and the number of visits on the prior year, and the number of previous PCR tests. So with all these, all these covariates were used to calculate the propensity scores, that it was like a logistic regression, and then we had like for each one of the individuals a probability between 0 and 1, and that is the propensity score. Then we started, so we have the propensity score. That was the first step towards our study. And now that we have our cohorts of interest, the propensity score, let's see which are the different methods that we are going to try to compare. The first one that we used was weighting. So what we did, like if you remember at the beginning, I was saying we have a problem with the index date. The unvaccinated don't have an index date. So what we did is to sort the index date of the unvaccinated following the same distribution of the vaccinated one. And then we used that to calculate the weight. So index date was part of the weight propensity scores too. And then we used 3 different sets of weights, with one with no geographic identifier, one with regional identifier, and one with GP. There is like the care site identifier. These are like variables that were also included in the propensity score. Regional identifier, there are 9 regions in England, so there are like 60 million individuals in England, so 9 regions, that's like an aggregation of millions of individuals. And then the GP is the care site, that is more like of thousands of individuals. So it's like granularity over the location that we use it in the propensity score. Why we cared about that is because, well, we think that in COVID-19 vaccinated related research, it's very important to account for different rollouts, and rollouts were not equally distributed in all the different GPs and regions, and the same for the infection rates. So we wanted to see which was the impact of these different rollout and infection rates in our propensity scores. And then we also used 2 different weighting methods. So we have on one side the inverse probability treatment weights, the IPT weighting, and on the other side, the overlap weighting. These 2 methods are different. They account for different things, and they have different formulas. So with this, the 3 combinations of different weights, depending on which is the granularity included in the propensity score for region, and the 2 weighting methods led to 6 different combinations that we will test. So these were like the 6 first methods that we tested in our study. And we tested a total of 8 methods. So the next 2 were related to matching. How we did matching? In the matching case, what we did was like we imposed an exact matching on age group using a 5-year bands plus sex and the geographic identifier. For the geographic identifier, as we did in the weighting part, we explored 2 different sets. So the regional geographic identifier, there is like these 9 big regions across the whole England, or this GP geographical identifier that is more like granularity around like thousands of individuals in each one of the care sites. And then we used 0.2 caliper matching using the nearest neighbors and the propensity scores. For the matching, we have the same problem that we had with the index date that we sorted in the weighting methods. We sorted sorting. In the matching population, how we do it, it's like we we calculated the propensity scores at the first day of the study. Everyone on the 4th of January 2021, and then we match individuals. Then once we have the pairs, we assign the index date to the unvaccinated, the one of their vaccinated pair, and then we recalculated the propensity scores. If the 0.2 caliper matching was not held anymore after the index date assessment, then we discarded that. We didn't consider that pair. So yeah, here on the right, we can see the distribution of the index date. As we can see in the weighted, we had more population than in the matched, and we are going to go now through the numbers. So yeah, and finally, outcome. Our study had 3 different outcomes. First of all, we had PCR testing, expecting that there would not be differences between vaccinated and unvaccinated in terms of PCR tested, and that if we would observe a difference of COVID-19 incidences, and it was not due to a lack of testing. Second outcome was a PCR positive test, and the third outcome, and it was a combination. It was like PCR positive test or a clinical COVID diagnostic. Our outcome model was like a quite often used method, the Cox proportional hazard regression model. So yeah, going around the numbers, as said, we started with these 40 million individuals. If we subset to the ones older than 75 and in observation on the 4th of January, we ended up with 1.1 million individuals. Most of them fulfilled the 6-month at least observation period, and most of them had the regional ID requested. Then we separated in vaccinated and unvaccinated, and we eliminated those that received a vaccine before 4th of January or the ones that had COVID before 4th of January. And with this, we ended up with our population. So it was like nearly 600,000 for the vaccinated cohort and a bit more than 300,000 for the unvaccinated cohort. Then for the weighted, what we did is like sort the index date of the unvaccinated and then check that there was no COVID-19 before index date. There could be like individuals, if we sort the index date on the 3rd week of January and they had like COVID on the 2nd one, like that could be an exclusion case. Also, we addressed that there were no record of death or left the database before the index date. And this is the population that we used for the overlap weighting analysis. So the 3 overlap weighting analysis used these numbers that are fairly more or like nearly the ones that we had at the beginning. Then for the IPT weighting, as we have trimming for like a 0. We want to eliminate extreme weight, so we only consider individuals who have a propensity score between 0.05 and 0.95. Then this led to different results for each one of the analyses, because like the propensity scores were different, including no regional identifier, the regional identifier or a GP identifier. And yeah, we see that like the more granularity we had in the GP identifier, we lost more patients because we had more extreme weights. And yeah, the last case, the matching case, we started with the vaccinated and unvaccinated. We did like a first pairing on the 4th of January. This led to 200,000 pairs for the GP matching and 250 pairs for 1,000 for the regional matching. Then we excluded again individuals who had COVID before the index date, a record of death, or left the database, and we ended up with these numbers. And these numbers are the ones that we used in the study. So then we have our populations, we have our methods, and our outcome. How are we going to follow them up? We followed them till the end of data availability. We had around 1 year of data availability. So the data cut was from end of December 2021, our study from January. So we had nearly 1 year of data availability. We censored them if they had like the outcome of interest, if like for the unvaccinated, if they were vaccinated, we also censored them if they left the database or they had a record of death. In the case of matching, so this only affects to the matching analysis, if the unvaccinated pair was censored, the vaccinated one was also censored, only if the censoring was due to vaccination. So yeah, now we have we've defined like our study populations and 8 different methods that we want to compare. So how are we going to compare? We are going to compare 3 things for each one of the methods. First one, to compare observed confounding, we are going to use standardized mean differences. Then we are going to compare power, and for this, like we are going to use the minimum detectable risk. And we are going to use finally we are going to measure the unobserved confounding, and to do that, we will use negative control outcomes. So we will compare the performance of these 8 methods across these 3 things. And so first of all, observed confounding, we use the standardized mean differences, and basically we impose the 0.1 threshold to determine if covariate was balanced or not. We can see the results in this plot. So I'm going to explain first how to interpret it. Like for this plot on the left, the x-axis is the SMD, each one of the points, it's a different covariate. Then the x-axis, it's the SMD before weighting, and the y-axis is the SMD of the covariate after weighting. So if the covariate is under this 0.1 line, it means that it was balanced after weighting. If the point is like more on the right of the 0.1, this means that it was unbalanced before weighting. So what we see is that we are able to balance most of the covariates that we were worried about. We see that we only balance for region if we account for that. So for the propensity scores where we didn't account for region, this, and even like IPT or overlap, we didn't balance because we were not adjusting for it. And then for the GP surgery, we've seen the same, that if we didn't account for that, we couldn't balance. And in fact, for IPT weighting, we see that even if we account for the GP surgery, we were not able to fully balance that, even if like it's smaller, but it's not smaller than the threshold that we forced of 0.1. For the matching results, we see similar results that we are expecting. SMG is quite small, everything balanced, only GP was not balanced for the regional analysis because we were not accounting for that. So take home message, the one that performed best, it was the overlap propensity scores with GP. That was not a surprise. Overlapping weights in principle are by definition like accounting for observed confounding. Then second metric that we use to compare the minimal detectable risk related to the power that we have. We see that for the unweighted, like for all the weighted analysis, the power that we have is quite similar to the unweighted one. And for the matching, we lose some individuals. We've seen in the diagrams before that we lost some individuals, so we have less power, and that was something expected. Then last metric to evaluate unobserved confounding, how we accounted for that. We said negative control outcome. So we say like a negative control outcome, it's an outcome that it's not expected to be influenced by the exposure, but shares like the same confounder structure. So we had 43 negative control outcomes that they were selected by clinical knowledge, and by definition of the negative control outcomes, we expect that only 5% would be significant. If we observed more than 5%, we used empirical calibration on the outcomes models to account for unobserved confounding. So the negative control outcome results, we can see that, well, for that was also expected. For the unadjusted, we know that there is like lots of observed and unobserved confounding. We see that 75% of the negative control outcomes are significant. Oh, yeah, let me explain first the diagram. So this diagram, the x-axis is the hazard ratio. So 1, it means that there is no difference between vaccinated and unvaccinated. If it's on the right, it's like more present in the vaccinated, on the left, more present on the unvaccinated. Then the standard deviation, it's the y-axis. So the more closer to the 0, the more power that we have to assess that outcome. The more far from it, the less power that we have. So as I was saying, and the unvaccinated, and then we have these lines that are the diagonal. This is what we would expect, that 95% of the point are within this triangle if the negative control outcomes are correctly balanced. So and we would expect only 5% to be outside. So in this unadjusted analysis, we don't see the 5%, we see a 76%. That's not surprising. We know that we have differences on age, on comorbidities, and lots of problems with the unadjusted. We were not adjusting for confounding. For the matching, we see that there is like an improvement. We don't see the 70%, but we see still like an increase around 40% on both of the matching techniques. Like 40% of negative control outcomes were significant. For the IPT weighting, we see that we have lots of power, but like many are significant. And again, we account for that with using the propensity scores and the weights, but we are far from the 5%. So we are still 65% of significant negative control outcomes. And finally, for the overlap weighting, are the ones that performed the best. It's still higher than the 5% that is like the ideal random, but like it's reasonable 20% of significant negative control outcomes. So yeah, so with this, again, overlap weighting is the one that performed better, and like in particular, the GP1 was the best one. So now let's go to the outcomes. We said that we would evaluate 3 different outcomes: PCR, PCR positive or PCR positive plus diagnostics. We see that, well, the unadjusted show like some and like some results that we are expecting, like the cohorts are unbalanced. So we see that, for example, the AstraZeneca populations showed more testing, whereas the Pfizer ones had less testing, and so. But then after we adjust for most of them, we see that we are able to reproduce these non-differences expected of testing between vaccinated and unvaccinated. And then we see that both outcomes, the only PCR positive or PCR plus diagnostics, are significant with fairly similar estimates between the different methods. So with this, our learnings were that overlap weighting was the method that performed the better. It was the one that minimized better the observed and unobserved confounding, and that region is very important for COVID-19 related research, and showed and like accounting for it, it's important on the propensity score. So our take-home message is that we will use overlap weighting with using the GP identifier. Then we conducted a target trial emulation study. I'm not going to enter much in the details, but we tried to follow the same structure that the AstraZeneca and the Pfizer trial had. And we used overlap weighting, and here we can see the estimates with and without calibration, and using as outcomes only PCR positive or PCR positive plus diagnostics. And yeah, so we can see that using PCR positive plus diagnostics, we can perfectly replicate the AstraZeneca trial and correctly reproduce the Pfizer trial, in fact, showing more effect than the one seen in the trial, but like overlapping in most of the cases. And finally, we also conducted a study comparing with the recommendation of, and there was an estimation of at 12 weeks to do like just one vaccine, use only one vaccine of Pfizer, and we compared this estimation with our results, and we see that we are able also to reproduce, and that our results with real-world data are in line with this estimation. So our target trial emulation had some limitations. Even we tried to emulate and follow the same patterns, there are some limitations on real world data that we cannot account for. So the first one is like some differences on the study population. For example, we focused our population on the older than 75 to account for the rollout of vaccination here in the UK, whereas like the phase 3 trials were done just in adults. And then the follow-up times in the trial, like they were follow-up for exactly 21 days where they received the second dose, whereas we followed them up till the 21st days, or we censored if they received the second dose before this day. Similarly for the AstraZeneca, where we followed them like after day 21 till they received the second dose, or the 12 weeks that they did in the trial. So yeah, and then learnings for the trial part, we used real-world evidence to successfully replicate the findings of the phase 3 trials. And despite of the lack of trial data, our findings suggested that the first dose of Pfizer that this estimation that was done by the Joint Committee of the UK of 12 weeks, our findings are in line with that. So yeah, all this is published in this article, Observational Method for COVID-19 Vaccine Effectiveness Research. Here you can find the link, and there are a bit more details of what I've been explaining and covering today. So now if we remember at the beginning of the study, we said, oh, we are going to do this study to evaluate different methods so we can answer the question of are vaccines effective against COVID and with real world data, and then how we can do it. And now that we know how to do it, we use those methods to evaluate other outcomes, for example, long COVID or post COVID-19 thromboembolic and cardiovascular complications. For time purposes, I'm just going to focus on the first application, but I'll give the links to the second one if someone is interested. So what we did in this long COVID study, we used 4 different databases, the same that we used for the method research, but also a second primary care data here in the UK, primary care data linked to hospital data in Catalonia, and the national health insurance claim in Estonia. The 4 databases had COVID-19 testing and vaccination linkage, and all those data were mapped to the OMOD common data model, so we only had to do one script that was reproduced across the different databases. So what we did was a staggered cohort study. As you can see, the first cohort is the one that we used for the methodological research. So this study cohort of age about 75. And then we used 3 subsequent staggered cohorts to compare vaccinated and unvaccinated, taking into account the rollout of the vaccination campaign. So so these were adapted in each one of the countries. Now I'm showing the one in the UK, and each one, and they were adapted a bit in each one of the countries to take into account the different rollout guidelines and their particularities. Then, we conducted this as federated analytics. So a common analytical script was developed and everything is in OMOP. We could adapt the data script in all the databases. We only had to account for the different inclusion criteria for the cohorts. And then what we use are the methods that I've just described before. So large scale propensity scores, including key variables, plus data-driven approach. The overlap weighting, assess the diagnostic we did with assessment of to covariate balance. We used negative control outcomes to address and measure confounding. And then we used fine and gray regression instead of the Cox proportional hazard ratio regression, and to account for basically competing risk of death. And finally, as we've seen, we are going to have for each one of the databases 4 different estimates, one for each staggered cohort. We conducted a meta-analysis of them. So going directly to the results and not giving more details, basically what we've seen is that we see that there is an effectiveness of the COVID-19 vaccine against long COVID. Around this, the effect is different in each database, but around 50%, and it's significant in all the cohorts. Only some of them that had very wide confidence intervals, we don't see a significance, but the meta-analysis, as I said, it was significant in all of them. And we also stratified those results by brand, and we can see that for both of the brands, they are effective against long COVID. So This study was published in Lancet Respiratory Medicine. Here I have the links of the Shiny app, where are like all the different results and, well, lots of more results than like just the figure that I showed, plus the link to the paper. Then we used we conducted a second study. This one was like exactly the same design, but changing the outcome to post-COVID-19 thromboembolic and cardiovascular complications. I'm not going to show the results of that, but again, I'll provide the Shiny app and the link of the paper. So if anyone is interested, you can read about it. And just to finish, one important thing and like a thing that we are very proud of, it was the reproducibility. So when we published the study that I explained about long COVID vaccine effectiveness, the code was publicly available, and then some data scientists in Norway, they were able to use that code, and with their database mapped to OMOP, they were able to successfully run the study and obtain, again, similar findings. As you can see, effectiveness around like 50%, and the 4 cohorts, again, presented like a reduction of the long COVID outcome. And again, this was published as a correspondence in the Lancet Respiratory Medicine. Here is the link if anyone is interested. So just finishing, this was like a team effort. Thank you to all my team, and especially to Annika, who had major contributions in the study design, with Danny, and also Nuria, who helped on the development of the code. So yeah, thank you very much, everyone, for your attention. I hope it was interesting. Here is my mail if anyone wants to reach out, but now we are going to have like 15 minutes Q&A. So yeah, thank you.
Right. Thank you, Marty. I see we have a question from Cindy. Cindy, do you want to come off mute and ask your question?
Sure. Thank you very much can you hear me okay?
Yeah.
Oh, wonderful. Thank you very much for your presentation. Maybe I missed it, but I have 2 questions. The first question is about when you sample the unvaccinated population, are those people allowed to receive COVID-19 vaccine doses at a later time, and then they will institute another index date?
Yeah, let me go through the slides. So basically, what we did. So, well, we account that like no individual should have like a prior record of vaccination. Then we separate the individuals in like, we use this 4 weeks of January, and we separate them on the ones that they receive a vaccine during those 4 weeks, and those who didn't receive the vaccine. And after that 4 weeks, if they receive a vaccine, is where censoring plays, well, it's taken into account. And I think I have, yeah. So we, if they receive a vaccine after that period where we create the groups of vaccinated or unvaccinated, if they are from the unvaccinated group, they are censored.
I see. Those unvaccinated will, once they receive the vaccine, if it's COVID-19 vaccine of interest, they will be included in the exposed population.
Yeah.
Or these people will be essentially censored and won't enter the cohort. So is it the former?
So we eliminate anyone who was vaccinated before the 4th of January, cannot contribute to any of the cohorts. Then we have this first 4 weeks to separate them in vaccinated and unvaccinated. And if they receive a vaccine after those 4 weeks, we use that to censor. Does this make sense?
I see. I guess the question is, maybe you mentioned it again. What is the rationale of setting the 4 weeks interval to check this vaccine history?
So we wanted to follow the UK guidelines for the rollout of vaccine. So during those 4 weeks, it was when it was recommended to, or it was available to vaccinate, start vaccinating people older than 75. We didn't want to be like a very long period. So because then like you have problems of like that we would not have unvaccinated. So we decided to restrict to just those 4 weeks according to the national guidelines of the rollout of the vaccination.
I see. Yeah, thank you. I guess I have another question if there are no additional questions from the audience. So my second question is, I noticed that the sampling that you mentioned, I think is a couple of slides before this one. So you assign the index state to the unvaccinated population based on the index state distribution of the vaccinated population. So I'm just wondering, have you ever compared the sample size using this approach versus you just do exact matching on the calendar date? So for example, if you have an individual who are vaccinated on October 1st, then essentially you look in the pool of the population who are not vaccinated on that exact date. And essentially you can just sample those people into the unvaccinated population.
So, yeah, so what we've done, and this is in future studies of that, it's to reduce this 4 weeks period to just 1 week. So we don't have like more, so we like shorten these timelines. And then at every week we see the ones that are vaccinated, the ones unvaccinated, and then you match them and assign the pair dates. I don't know if this answers.
I see. So it's kind of like a nested trial design. So essentially, instead of looking at the exact single day, you are looking at a week interval. And I guess the question to you is, are you going to assign the index date as a start of the week, or are you going to assign the index date, I guess, in the middle or a random date within a week?
So like this was like in studies after that. So like this weekly matching was not done in any of the studies that I've presented, but then you assign the date to the first day of the week. You
first
do
of the week. Okay.
what we did here, this process, but just with 1 week instead of with 4 weeks. So you assign the date at the first day of the week. You then find pairs. Then you assign the unvaccinated one the date of the vaccinated, and then you recalculate the propensity score. And if something happened in those 4 or 5 days, then you remove that pair.
I see. Yeah. Thank you very much.
Right. George, I know you have a question.
Hi, Marty. Thank you so much for the talk, which was so clear and easy to follow. So thank you for that. A couple of questions. First, I think, the first week after vaccination is not some see it as not effective, so it should be a zero effectiveness. Others see it as hyper effective because you mistake your COVID symptoms for vaccination symptoms and you don't get it diagnosed. In your study, you included the first week, but you think it just kind of gets washed out in the rest of the year that you're looking at the person. Is that what happened, or did you start a week after the vaccination?
So that's one of the limitations in our study. So when I show these estimates, these estimates, they include the first week. And we've seen that we were expecting, well, I think if you have in mind the trials, it's like they go parallel the lines and they separate after 1 week. We didn't observe this effect in real world data, and that was one of the limitations of the study. And there may be like many reasons behind that we could discuss a bit if you are interested, but yeah.
Okay, very good. Well, in our observational study, we also didn't see the two lines parallel, and we did chart review and showed that, in fact, what was happening is people weren't coming for their COVID test when they had symptoms because they thought it was due to the vaccine. So we actually had very high effectiveness in the first week, which wasn't true effectiveness. My other question, a second question is, in preventing long COVID, is this among people who had COVID? In other words, are you just showing that the vaccine stops COVID and therefore it also stops long COVID? Or are you showing that among people who get COVID, fewer of them get long COVID? Like what's the denominator?
Like the first one. So it's so basically we perform the same study that I was showing with COVID, and we changed the definition to COVID plus symptoms. So when we see this, there is two effects, one preventing COVID itself, and then preventing COVID plus the symptoms. So the denominator is like the whole population. It's not within COVID population, it's within background population.
Okay, thank you. And third, just to point out, and this is what we see also, in your negative control outcomes, in your best one, you had 21% false positive type 1 error rate, which is what we see also. Like, in other words, you would hope to see 0.05, and you're seeing a 0.21, which is why you do the calibration. It's not because you did a bad study. When we do studies also in other areas, not just vaccinations, we also see this higher than 0.05, and that's why we go ahead and do the calibration. So I was just kind of, the bad one was 76%. 76 is a bad study. When we see a 76, we know we're in trouble. But when we see a 10 to 20, that is typical of observational studies, and that's why I think you do the calibration.
Yeah. So we also observed in other studies like similar results. In fact, all the long COVID ones and like the subsequent studies that I've showed, all like the negative control are also reported, and the results that we observe are always between 10 and 20% if we use overlap weighting.
Which had impressive results. Thank you for that too, to see the success of that technique. That's it for me. Thank you, Maura. Thank you, Marty.
Thank you.
All right, and we have another question from Patrick.
Thanks, Maura. Thanks, Marty. Just to echo George's point, I thought your presentation was extremely clear. You did a really great job of going through quite a lot of work that's fairly complex in a very easy to digest way. So thank you for that. Towards the end, Marty, you somewhat nonchalantly said, "Oh yeah, and we went ahead and replicated this in Norway." And I know you didn't have much time to go through that, but I just wondered whether you could share a little bit more about the practical logistics. I think it's a remarkable accomplishment. You started with the UK and Spain, and then you went to Norway, where that perceptions about vaccination regimen, outlay, just attitudes, not to mention data issues about how does one even identify COVID in data. All of these things can vary substantially across geographies and jurisdictions. So I just wondered whether you could just maybe share a little bit more about the practical logistics of replication. What did you learn that you were surprised by, either positively or negatively, and what does this study have as like a broader implication for the research community about the role that replication can play in this type of work?
Yeah, thanks for the question, Patrick. So yeah, basically, so the original idea was to include, so Norway reached out. They know that we were doing that study, and they reached out like to participate, but they didn't have like their OMOP instance ready. And they said, okay, just tell us when you are ready and we will add you to the study. And then we finished the study and we published it. And then they said, oh, we are ready. And then basically we told them, okay, everything is like open source. We've published the study, but it would be nice if you can replicate it. So the team in Norway with quite different databases you were like describing, they basically run the same code. Again, they had to adapt a bit all these rollout groups because the rollout in Norway was different than the UK, than the Spain, than to Estonia. And yeah, like I think it's like a remarkable result in terms of reproducibility. So we've done a study using 3 databases. We published the code and someone else with a bit of our help, but they are able to run and obtain like comparable results, which yeah, that in terms of reproducibility and transparency, I think that it's very nice. And that's the story how we ended with the results with Norway.
So just to follow up there, Marty, you said like one obvious thing you had to change was you had to change the calendar times and the strategy, because that's of course like a country policy. So you wouldn't expect that to be reproducible, quote unquote. Were there other aspects of the study that you had to modify, like exposure definitions or outcome definitions or adjustment strategies or anything? Or for the most part, was that able to take advantage of the open source package that you had already prepared?
Yeah, so for all the rest, what we did is to make sure, like at the beginning when they said, oh, we want to run the same, we ran some sort of diagnostics to make sure that our definitions of the exposure and our definitions of the outcome were capturing correctly in their OMOP instance. And once we make sure that it was the case, then they were able to run the same study. And as said, we only had to change the rollout and like the staggered cohort design to adequate for the Norway rollout vaccination campaign.
It's terrific. Thank you, Marty. Thank you for your presentation and for all the great work you're doing.
Thank you.
All right, so we've come to the end of the hour. So I just want to say, yes, thank you again to Marty for presenting your work today. And thank you everyone for joining. Our next seminar is going to be November 20th, and Jennifer Nielsen will be presenting. So thank you everyone for joining today.